All right, welcome to part 2 of chapter 7, where we're going to look at crossing over and then errors that can occur in meiosis. All right, so the purpose of meiosis is, number one, to make gametes, but it also allows us to change allele linkage or mixing alleles. And there's two ways this can occur. Number one is crossing over, which occurs in prophase 1, and we'll go into that in more detail in a second. And the second is the random assortment of the sister chromatids that occurs in metaphase 1. We don't know to which end each pair of sister chromatids is going to go. All right, so crossing over is a form of recombination between non-sister chromatids of homologous chromosomes. All right, and what this does is it allows us to produce new linkages or new combinations of the alleles from the parent. Okay, this is a tetrad. This is a pair of sister chromatids from the blue chromosome, a pair of sister chromatids from the red. One of these, either the blue or the red, came from one of the parents, the other came from the other. Okay, and you can see the alleles they have. When you see capital letters, that's usually meant to be a dominant allele. I'm hitting the wrong button on the mouse, and it's causing things to move when they shouldn't. But you can see the capital letters generally mean a dominant allele, whereas the lowercase mean a recessive allele. So the blue chromosome only has dominant alleles. The red chromosome only has recessive alleles. And wherever one of these blue ones goes, it's going to get those 3 dominant alleles. Wherever one of these red ones goes, it gets 3 recessive. That's the combinations that we have. But what happens in crossing over is the chromosomes of the tetrad cross over each other and then briefly break and reanneal. It's like if you were stretching and the person next to you were stretching your arms and your arms crossed and they briefly broke and reannealed and you had their hand on your arm and they had your hand on their arm. Well, that's kind of what happens in crossing over. And see, what it just did was it created different linkages. Now, instead of all dominant alleles, I've got 2 dominant and a recessive. Now, instead of all recessive, I've got 2 recessive and a dominant. And so this is just another way to increase diversity because we're changing the allele linkages or the way alleles are inherited together. Okay, so again, sexual reproduction, we're fusing gametes from 2 different parents, and gametes are haploid, right? They arise by the division of immature reproductive cells, the germ cells, and in humans, that's the oogonia, that's the spermatogonia, okay? At fertilization, we fuse the gametes to form a diploid zygote. That's the first cell of the new individual. And now mitosis is going to kick in many, many times to produce a mature organism. But sometimes there are errors in meiosis. Probably one of the most common is non-disjunction. Non-disjunction occurs when either too many or too few of the chromosomes migrate. They don't separate properly during meiosis. And so what you get is a gamete that's either missing a chromosome copy or has 2 chromosome copies of the same chromosome. Okay, so if you're missing one, you're called monosomy. If you've got an extra, you're called trisomy. Okay, and this is what causes Down syndrome, trisomy 21, an extra copy of chromosome number 21. You can also have duplications occur, where a section of the chromosome is duplicated. So now for whatever genes are in that region, you've got multiple copies of them. You can have deletions, where part of the chromosome is lost. It breaks off or it's damaged and lost. You can have inversions, where a sequence of alleles will break and flip upside down and reattach in the wrong order. And then you can have translocations that occur, where the tip of one breaks off and attaches to the tip of another chromosome. Non-disjunction is very common. This is what causes, here's what happens. Here's the tetrad, and instead of separating like this did, these went the same direction. And so now when we go through meiosis 2, now we're going to get an extra copy of the long chromosome in these 2 gametes, and we're missing a copy in these 2 gametes. Okay? The same thing can occur here, where in meiosis 2, non-disjunction can occur here where both sister chromatids go the same direction. And now again, we got an extra and we're missing one. Okay, where we really see this is Down syndrome. And Down syndrome, the extra 21st chromosome is very much related to the age of the mother. If we look at, incidence of live birth percentage, so number out of 100, it's pretty low in people in women who are young, right? 20 to 25, you're probably, this is less than 0.75, so this is maybe less than 1 per 100, very, very low. And then as you age, now all of a sudden we get to age 40, and we get higher than that to 45. Look at the big jump between 40 and 45. Why is this concerning? Well, a lot of women in America are waiting to have families, if they have families at all. And a lot of that is because, those of you who are in college and are raising a family know how hard it is to do that when you're young, to do both. And so many, many women are putting off having a family until they've got their career underway. And of course, if you wait until you're 40, 45, it used to be, most women didn't wait that long, but it wasn't unusual for women who had a lot of children to still be having children in this age group. And then now all of a sudden they've got a Down's child later in the family. But realize that healthcare is different now, and so it's not as life-threatening for a woman to have children when she's older. So we're seeing an increase in the number of Downs. Now, is that bad or good? Down's children are different, but between you and me, I've never been around a Downs individual and they were sad. They're some of the happiest people I've ever been around. Granted, they have a shortened lifespan, and there's some other issues they're going to deal with, but who's to say being happy isn't a good trade? I don't know. Here's an example of an inversion. If you look, you can see, the bigger band, the middle band, the tiny band, and now the tiny band, the middle band, and the bigger band. The bigger band is in a completely different place, right? Well, if crossing over occurs, we can end up with a different arrangement of alleles. In translocation, we swap. The tips of one chromosome go to the other. The tip of the other goes to this one. It's kind of like crossing over, but not between sister chromatids. We can see these damage to chromosomes on a karyotype. Now, this is a normal karyotype. This is normal for a human female because the X are the same. But because we can match up the chromosomes and we can stain them to show similar regions, we can quickly identify if there's damage in a chromosome. X inactivation. In mammals, females have 2 X chromosomes and males have an X and a Y. But in females, a female doesn't express the genes on both the X chromosomes. All right, what does it mean? When you express a gene, you use the gene to build a protein. But if you've got 2 X chromosomes, you've got double the genes that are on an X chromosome from a male. If you express them both, you'd have double the protein production. So what usually happens in a female animal is one of the X chromosomes is inactivated. But that individual doesn't know which X chromosome is going to be inactivated. And so you can get different characteristics depending on which is inactivated. And this is a tortoiseshell cat, which gets its coloration from different chromosomes encoding different colors. One of them got inactivated. Deletions, Cri-du-Chat syndrome, is a nervous system abnormality. There's characteristic facial features, but what it's named for is the high-pitched cry that infants make when they have this. And this is showing you the same individual from ages 2 to 4 to 9 to finally 12, right? And again, this is because you've lost most of the small arm of chromosome 5. So next time, we're going to break into Chapter 8, and we're going to look at patterns of inheritance, and we're going to find out why you can have, a family. You can look very much like your siblings or nothing at all like your siblings, but still share the same parents.